Yes. So retroviruses, which is already done to you, enveloped single-stranded RNA containing animal viruses that carry an enzyme reverse transcriptase. Isn't it? I did this all, but I want you to remember reverse transcriptase and envelope. That means they have envelope and they have single stranded RNA as genetic material. So, generally, when we read about retroviruses, we tend to think what are the differences between retroviruses and bacteriophage, that is the virus which infects bacteria, because that is the most which we study in even learning lytic cycle and lysogenic cycle. So I am going to do differences between retroviruses and bacteriophages, which are viruses which infect bacteria. So you can see here, whichever is in blue color is retrovirus, whichever is in green color is phage. Any virus that belongs to Retroviridae. So what is the name of the group to which retroviruses belong? Retroviridae. And any group of viruses that infect bacteria is called as bacteriophages. They have single stranded RNA as genetic material. But phages may have DNA also, RNA also as genomes or genetic material. So my major point is not every virus that has RNA is retrovirus. Okay, then what makes retroviruses different from other RNA containing viruses? They have an enzyme called reverse transcription, isn't it? So contain their own reverse transcriptase enzyme, whereas the other viruses, even though they have RNA as genetic material, does not have reverse transcriptase. That makes Retroviridae very unique because they can perform retro transcription, that is transcription in opposite direction, because they have reverse transcriptase enzyme, whereas other viruses which has RNA as genetic material cannot be retroviruses because they don't have this enzyme. Then you may ask me, what is the point? Which cells they will infect? They will infect the cells which has RNA as genetic material. But retroviruses can infect the cells which has DNA as genetic material because they can perform reverse transcription. That makes them unique. Then RNA genome is reverse transcribed to DNA, which permanently attaches to the host genome. They exhibit either lytic cycle or lysogenic cycle. We did this. They attach to the plasma membrane proteins and glycoproteins. Tail fibers attach to the cell wall proteins. Capsid enters the cell, whereas genetic material is injected. So if this is the envelope, this is the capsid, this is the RNA which is genetic material. I am speaking about retrovirus. this is envelope, this is capsid, this is genetic material. So the envelope merges with the host cell membrane. So it merges with the host cell membrane. The capsid along with RNA is injected inside. Whereas in case of phage, what happens if this is the host cell membrane, then the virus will land on the host cell by means of tail fibers and only genetic material is injected inside. So here capsid along with genetic material is injected because it has envelope. Whereas the phage bacteria only genetic material is injected. Biosynthesis occurs in the nucleus in DNA viruses and in cytoplasm in RNA viruses. Synthesis occurs in cytoplasm. They are speaking about assembly. Enveloped retroviruses are bud out. Enveloped retroviruses are bud out and non-enveloped will rupture the plasma membrane and released from lysis of the host cell. So these are the differences between bacteriophages, which can have both DNA and RNA as genetic material, and retroviruses, which specifically have single stranded RNA as genetic material and with an enzyme called reverse transcriptase.